Well, welcome to Progenesis Academy webinar series. Today, we are very pleased to introduce our speaker, Klaus Wiemer. Dr. Wiemer holds a Ph.D. in reproductive physiology with emphasis on embryology. He is the laboratory director for POMA fertility. And we have a wonderful panel with us, including Klaus. We have Dr. Levent Keskintepe, who is currently the laboratory director for LA IVF, among other labs. And we have with us Dr. Dmitri Dozortsev, who is the laboratory director for Advanced Fertility Center of Texas. And he is the current president for the American College of Embryology. Today, the talk will be about returning to operation following the SARS-CoV-2 pandemic. Klaus, thank you so much. The floor is yours.
Thank you so much for the wonderful introduction. I am pleased and really honored to be here. We're going to talk about returning to operation following the SARS pandemic. So this is my disclosure. So just to get started, so really, as we all know, this is just a few very obvious things for all of us, and that we exist, in the current pandemic that has gripped all of society in so many ways. And so IVF laboratories at this point in time have all been shut down at varying degrees, whether they shut down immediately or were able to finish their IVF cases that they did. The point is many labs have been closed down much longer than they thought. So I thought that I would take this opportunity to discuss these four major topics: cleaning the exterior of the laboratory, and I'm going to diverge a little bit to talk about some viral transmission that's potential mostly in large animals, because I think that's a good model. I want to talk about our HVAC systems that exist today, because I feel like HVAC systems could potentially be an area that we can have ongoing contamination. Then we're going to talk about how do we clean the inside of the lab, what are the areas of concern, and then QC process to ensure that the lab is fully functional, and then how do we reopen the labs today at our centers today, and doing it as a good stewards and good citizens. So really, my goal today is really to provide some suggestions that could be used really anytime. So in other words, I'm hoping that I could provide some suggestions for people that maybe have not thought of them. So we're going to get pretty organic in certain areas. If this is something that you already know about or know a whole lot of, I apologize. But really, the whole goal of this is to try to take this time and try to reinvent the processes that we're doing and reinvent what we do. And then including with that, we're going to talk about quality control, how to improve our quality control processes. So as we all know, this beautiful little virus has been creating a lot of havoc for us. It's a single-strand RNA virus. I'm not really going to get into it, what it is, because we've had some amazing lectures earlier about that. But what I want to stress is that there's limited ability of this virus to bind because it requires an ACE receptor, ACE2 receptor, but this receptor site has been found, as listed in one recent paper, in some of the testicular tissue, as well as found in developing spermatogonia. But up to this point, we've really had no viral transmission or no vertical transmission yet. So I want to talk a little bit about the Zona Pellucida, because this is a structure that all of us in the laboratory at least know what it looks like. But really, I thought might be a good opportunity to revisit what is the Zona? Well, the Zona is really this complex structure that's made up of 3 sulfated glycoproteins. The Zona really acts as a wonderful barrier due to its acellular nature, its thickness, and the lack of receptor sites that are present on there. What's interesting though, once you look at the Zona from an electron microscopy perspective, you see that it's very spongy-like, that it's a complex network that has many, many pores, and these are the pores that existed are remnants of how the granulosa cell is able to communicate with the oocyte prior to ovulation. So what happens is that these pores decrease in size as you get closer towards the interior aspect of the oocyte. And these pores change in the zona dramatically following ovulation and fertilization. So the information that we know about pathogens crossing the zona pellucida is really very, very low. So this is an electron microscopy imaging of 3 zona pellucidas. We have the bovine zona pellucida, and what's interesting here in the porcine and the human, what's interesting is that the size in the holes of the zona, this is the exterior aspect of the zona, range, 200 to 300, 400 nanometers in the human to almost 1,000 nanometers or 1 micron in a porcine oocyte, whereas in the human, these pore sizes tend to be all under 192 or under 200 nanometers. If we look at the viruses though below, you'll see that a bovine BVD virus, this virus can be as small as 60 nanometers all the way up to the size of the human coronavirus that we're dealing with is the size of 120 nanometers. So we see that the virus that we're dealing with right now is a pretty large virus. Now this is really good because large viruses are much easier to remove through centrifugation than smaller viruses are. But I just wanted to show you that what the zona pellucida looks like. It's just not, a bunch of holes in a zona. It's a fairly complex architect in comparison to the viruses that are commonly found in these host animals. So why are animals a good model for all this? Well, really, the reality of it is there's very little data that exists in human studies. But we can all take great comfort in knowing that before 2005, there were many, many embryos that were transferred in donor oocyte models, and we had no known diseases transferred. There are some literature sightings of HIV found in poorly prepared semen samples, but that's really what we know right now with humans. So really, if we want to now take some time and learn about this, studying animals is really probably one of the best models to study as far as how it relates to humans. And the reason for that is that we can take these animal embryos, expose them to pathogens at different levels, and we can transfer these embryos to recipients, or we can take animals that are serologically positive for certain viruses, mate them, transfer those embryos to serologically negative recipients, and see what the outcomes are of that. And in one of the first papers, it was Singh et al. 1982, this author worked at the time at the Agricultural Research Center up in Canada, which has done a marvelous job along with Stringfellow and other authors to really study the effects of viral and bacterial transmission, the potential of that through embryo transfer. Well, in this particular study, which is kind of considered a landmark, they were taking embryos at various stages from 16 cell to the blastocyst stage. We're exposing them to various levels of this virus, the IBRV, and there was no evidence that the virus could be transmitted through the zona. And what was found, interestingly, and this is one of the first papers talking about the concept of washing the embryo, and that washing the embryo effectively removed this virus from the zona pellucida. Embryos that were collected from serologically positive donors that were transferred to recipients also were negative. So this study showed that the virus could not infect the embryos and could also be removed and was not going to infect the recipients. And this kind of theme exists with the next one. So I thought, okay, well, viruses are big. What's the next smallest thing we can look at? So now we looked at the prion that causes BSE. And this paper from Rathall et al. And veterinary records show that basically they were not able to transmit BSE to recipient animals. These recipient animals were allowed to grow for a long time. The babies that were born also did not exist. That they did necropsy of the brains and never did find any evidence of this virus at all in any of the spinal fluid or brains of any of these animals. And then last but not least, I chose the goats because the goat the embryos are collected surgically. The ability for there to be a lot more fluid transmitted and stuff like that is kind of maybe more or less looks more like what we do in an IVF lab, although not exactly. But the interesting thing is these embryos were collected, they were frozen, and then transferred to CAEV-free recipients, and the offsprings as well as the recipients were negative to this virus. So, you know, you can look at a lot of papers and find that the vast majority of the literature states that there's really not a lot of issues with this. So the studies really seem to indicate that the virus doesn't readily transmit, and that's probably due to the lack of receptor sites as well as to the actual physiological structure of the zona, as well as the changes that occur to the zona pellucida following ovulation and fertilization. And then we know, because in the embryo transfer industry with cattle, that viruses can easily be removed from the zona pellucida. And in fact, this is something that's required of all embryos prior to them being frozen. Now, one thing that has come up in reading these papers are that if there was a theoretical method of transferring a virus to an animal was by transferring hatching blastocysts or zona pellucidas that are compromised. So maybe, you know, maybe we need to maybe we should go back and revisit not using ICSI so much, and perhaps we could go back to doing a little bit more conventional inseminations. Again, this is an example of where we could maybe rethink things. So these are my own personal recommendations, but I have to tell you that it's out there. We have a paper that was published in Cryobiology that theoretically a liquid nitrogen tank can be the source of contaminants. We have found spores in virus, or I should say in the vapor phase. That until we understand this virus better, I would suggest trying to freeze oocytes, embryos in closed devices. If need be, we found out that this virus was indeed present, we could always develop enzymatic cleaning like trypsin, which is used a lot of times with cattle embryos. And then maybe for the time being, to be super conservative, that maybe these gametes should be quarantined until, you know, we decide as a society what's best. So now we're going to turn our attention a little bit to HVAC systems. So, and I'm going to break HVAC systems down into two basic types: closed systems and open systems. Closed HVAC systems are the ones that we see a lot more now. They're characterized by really high air exchanges, elaborate air filtration systems, and positive pressure relative to the rest of the clinic. Whereas open systems are found more in older settings, a lot of times in office IVF lab settings. What these all have in common, though, is that the ductwork is shared, you have limited air exchanges, and you have minimum air filtration, and usually pressure is neutral relative to the other rooms. So if we compare and contrast, closed HVAC systems have very robust ductwork because they have to handle the increased pressure. They also have advanced air filtration systems. So carbon and potassium permanganate is a hostile medium, I should say, for viruses. So it tends to kill the viruses. And the air exchanges are usually 16 to 18 hours air exchanges per hour. So we're going to very effectively disrupt any theoretical aerosol that's present in a laboratory, for example. And the positive pressure that you have in these rooms is going to reduce the potential for contaminated air to enter. So really what it's saying is that the theoretical viral introduction is greatly reduced in these kind of places, in these kind of scenarios, because labs that are built with these type of HVAC systems are also built for self-isolation so they can have positive pressure. So what about open HVAC systems? Here, they tend to have shared ductwork with the majority of the building, so there's many sources of contamination. Since the ductwork is not sealed because it doesn't have to handle high pressure per square inch, these ductwork can be a source of leaking. We have minimal air filtration also, so they tend not to kill the viruses. And the air exchanges an hour are typically less than 12. So that's really not sufficient to remove the aerosol that the lab staff are going to produce. And the other thing about it is these rooms tend to lack positive pressure. So the potential for outside air to enter the lab every time you open and close the door is great. So what can we do as far as just how to compare and contrast our labs as far as the HVACs go? Well, on the left are the closed system and on the right is the open system. So a lot of these things we discuss, but really these labs are best because they're going to literally scrub the air. They reduce outside contamination. They really provide the proper air exchanges that laboratory staff really need. And they provide positive pressure, which also decreases the amount of contaminants that can come in from the outside. So what can we do to improve this kind of HVAC system? Well, we can be cognizant of changing the filters because exhausted filters are not going to filter the air and therefore not going to remove viruses. And the other thing that we can do is change our scrubs every day because a lot of people seem to wear scrubs multiple times. They wear them home, they wear them back to work, and now you're introducing a source of contamination in a closed system. What about an open system, though? There's really a lot of things that we can do here, though, to help out. Before you inspect it, though, I suggest that you shut the system off and that you wear, PPE and gloves when you're doing this, because there's a lot of sources of contamination. So some of the things that you should look at, try to improving is like, well, upgrading your fan motors if you can. And realize if you do have HEPA filtration in these scenarios, they don't help. You need to install nanofiltration. In other words, where you're filtering down to the nanofilter. If you do that, though, it's going to require quite an upgrade in fan motor to deal with the added resistance that you're going to get. If possible, do that. If you can't, then try to do things like increase your positive pressure. That's going to often mean doing things like balancing the air in the laboratory. Increase your air exchanges an hour so that you remove aerosol as quick as you can. Replace these filters as often as you can. And make sure you clean the diffusers with something like a 70% ethanol or a 6 to 10% hydrogen peroxide. The other things that you can try to do is reduce staffing in the lab because, the air exchanges are going to be if you don't do that, then this could be a potential source of contamination. And like I said earlier, change your scrubs. So now we're going to talk about cleaning the laboratory, and the approach that we're going to take here is we're going to start on the outside and work our way into the laboratory. And my thoughts on this are really, you want to use multiple cleaners, and this is going to have, and the reason why multiple cleaners are that you're going to wipe the area more than once, so you're much more likely to get, any potential contaminants, whether they be viruses, molds, or yeast. But it also depends on what the surface material is and the sensitivity of the surface materials. Not everything does very well, for example, with hydrogen peroxide. So obviously, where do we start? Well, we should start with our doors and work our way inward. In here, I've kind of made some suggestions of what you can use. So we use these sandy cloth wipes that are a germicidal wipe that contains alcohols and ammonium chlorides because when these break down, they leave the lab relatively quick. We don't have any embryos in the lab, so we're not really worried about these latching on to any oil that we have in the lab, oil culture dishes. You also really need to clean the floor as well. And again, you can use something that's a combination of alcohols and ammonium chlorides. But again, it's really important that you allow these materials to sit for at least 1 to 2 minutes because it's a contact that kills the virus or the yeast or the bacteria. Hydrogen peroxide works great, but if you're going to do that, make sure you check the floor first, and by all means, wear goggles and a mask. Another area that we often don't clean as well as we should are the collection rooms. And using a 1% 7X solution, this is a detergent that's used very common in tissue culture labs and used in a lot of embryology labs because it's relatively toxic, less toxic to cells. And you want to follow by water on clean materials. So, you know, couches, things like that, clean very well with 7X, but they don't do so well with ethanol or hydrogen peroxide because it tends to discolor them. So those tend to work better on resistant surfaces. But again, the theme of it is you got to let the material sit there at least 1 to 2 minutes. Preoperative rooms, recovery rooms, these are all areas that don't tend to get cleaned as much as they do because we think they're outside of the critical care area. Well, now during this downtime is a really wonderful time to clean these areas as well. And you can use hydrogen peroxide again. One thing I do want to state here though is regardless of what cleaning agent you decide to use, I would suggest that you use a spray product whenever you can. And the reason for that is that the surface stays wet longer. So if you do use products like wipes, you're going to have to use them multiple times because you're going to have to you need to calculate for a 1 to 2 minute surface contact in order to, you know, effectively disinfect your lab. So that leads into the next thing. When you read tissue culture papers, they make a very clear distinction between cleaning a lab and disinfecting the lab. I'm going to kind of use those terms interchangeably, but I am going to tell you this: you cannot you cannot disinfect a dirty lab. So clean the lab first before you try to disinfect it, because all you're going to do is disinfect dirty stuff. Like I said earlier, change the HVAC system off if it's a open system. Think about the gloves that you're wearing to clean with. A lot of the cleaners that you may want to use may not necessarily work with the gloves. You don't want to do anything to your hand. These are things that people don't think about until their hands are burning. Clean from the top down. Light fixtures are really effectively cleaned with ethanol. How do you clean your walls? This is a great question. I'm not really sure how to answer that question because a lot of that's going to depend on is it an oil-based or a water-based? If it's water-based, you really can't use anything on that or else you're going to discolor the paint. If it's an oil-based, I hope you don't have, but on that surface, you can use a lot of different cleaners. The floor should be cleaned again, just like I stated in earlier. Again here, now that you're actually inside your facility cleaning, you really need to think about what to clean with. Don't use glutaraldehydes. And the reason for that is that glutaraldehydes are really effective cleaners. In fact, glutaraldehydes are used a lot for cold sterilization. The problem with glutaraldehydes is that they break down and they form these radicals that are extremely embryotoxic, and they persist for a long time in the air. And in fact, in tissue culture labs, most labs don't use them because the glutaraldehydes end up impacting cell culture development after cleaning. So please don't use glutaraldehydes. After you turn your HVAC on, allow it 1 to 2 days to completely clean out and get new air in there. If it's an open system, if you have a closed system, overnight is more than enough time. So now that you've cleaned the lab and now you're standing there, the next thing I would do is kind of look around at your equipment and go, okay, what's the highest potential? What's my lowest potential? And water baths, box incubators, circulating water bath, incubator fans, all of these things are areas that have high potential. And these should be cleaned as I've listed here in the slide, because these are great sources of bacterial and fungal contamination. So, this is time to do that. Hoods and surfaces tend to be lower potential for contamination. So we tend to clean those with 70% ethanol, followed by hydrogen peroxide. And again, we're letting it sit. And something that's very common in tissue culture labs, but not so much in embryology labs, is use overlapping motions. When you clean with one solution over another, work at 90-degree angles so that you always have overlap. Change your hood filters. Benchtop incubators are really easy to clean. If depending on what type of water reserve they have, you either replace it or clean it, sterilize it. If you have a plastic water reserve, make sure you fill it with water, let them off gas, because many times you will fail a mouse assay if you don't rinse and off gas these bottles. Centrifuges are an area of great high potential. And the reason for that is centrifuges don't tend to get cleaned as much as they should be. If you do clean your centrifuges, though, make sure you don't use alcohol on the rotors. That's a big no-no. And clean the inside of them very, very well. I clean them at least 2 times with hydrogen peroxide because these are a great source of contamination. What about other things in the lab that we touch all the time? These are things like injectors, control pads, computers, scopes. Depending on what the material is, either clean it again with hydrogen peroxide or ethanol. And we tend to do it twice, just to make sure that we overlap. Don't forget to clean the eyepieces on your microscope and knobs. This is something that people don't do. If you were to look at eyepieces are a huge source of contamination between staff members. So eyepieces should really be cleaned at the end of every day. Other things like buttons, flasks, phones in the lab, diamond pins, anything that you touch all the time, probably wouldn't hurt it to give it a good cleaning now as well. So my final thoughts on cleaning are: start with the exterior, work your way down, start at the top. If you must clean, you must clean before you disinfect. Use multiple cleaners, that's what works best. Sprays are more efficient. Concentrate on the equipment that has high humidity levels or water could potentially be standing. Make sure you allow your lab plenty of time to off gas. And that's going to be dependent upon your HVAC system. And also know that your HVAC system moving forward could be a source of contamination. So as I stated earlier, I wanted to take this time to not only think about how are we going to open again, but what can we do to reinvent ourselves? So reinvent our processes that we use in the lab. And one of them is quality control. So an area that I think is very important is that let's start with our surface temperatures and our culture drop temperatures. Who knows when's the last time you actually did that? I think what I would want to offer to you here is to go through your mock procedures. Make sure that the culture media does not raise too high and err on the cool side. Now's a great time to do QC on your gases and also develop a tracking system for your consumables. And then when you're done with everything and you feel like the lab is ready to open, this is a wonderful opportunity to develop an increased sensitivity mouse assay so you can make sure that your lab is ready to open. So here's just a few things that we do in our lab. The slide here on the right will show the template of, for example, of a 6-foot hood. So when we go to start QC, we check all the places in the hood where people would potentially put dishes. And then we actually go through mock procedures. And I've given some suggestions here at 0, 5, and 10 minutes. You want to place the dish under the bubbler because you want to actually recreate the events that you're doing. And the reason why this is really important to do is because a lot of us use dishes with different architect on the bottom of the dishes. So some of them have flat bottom and others have raised bottom dishes. And so that's actually going to impact temperature much more than you think. So the take-home message here is really mimic the procedures that you do in the lab. And that's the same thing that we do with our microscopes. Again, I don't know when's the last time you calibrated your heated stage, but we all know that many of them have notorious poor thermodynamics. They heat cycle and can make it worse if they're open and closed. So again, you want to check for hot and cold areas. And again, when we're doing these things in our laboratory, we tend to mimic processes that we're going through in order to see what our temperatures are. So on the right here, our hood, or our heated stage has the outer areas heated as well as the inside. So we check all the outer areas for hot and cold spots. And then the interior, we also check for hot and cold spots because we want to make sure that when we're doing ICSI or things like that, that we're not actually getting too lethal with our temperatures. So you would be surprised what oil over the top does to your temperatures. So again, now let's assume your lab has been closed for a while. So if it has been, this is a really good time to flush your gas lines and flush your filters out if your incubators have been off. You may even consider changing the filters that are in your laboratory. And the reason for that is that these filters have accumulated VOCs. So when you turn the gas on, depending on what the VOC load is on these filters, you could actually release a whole lot of VOCs into your lab. The other thing you might want to consider is upgrading the quality of your CO2 and nitrogen, mostly because CO2 is the most contaminated gas in your triple gas mix, and nitrogen because it's the largest percentage of gas in your gas mix. If you have incubators that can mix their own gases and you have your own manifold system, consider using the liquid nitrogen vapor. It's extremely clean, much cleaner than you would ever find in a cylinder. Perform pH testing once your incubators are fully functional, and incorporate pH testing with every new lot of media and gases you get. Consumables, we talk about this a lot, but a lot of people don't have a good inventory system. But buy MEA products whenever you can. Develop sperm assays whenever possible. And our sperm assays are long exposure. In other words, we expose the material in question for up to 72 hours before we actually do a sperm survival test. Off-gas your products. Track when everything comes into place. And if you can, hold lot numbers, because this will really help in your cost, and it'll reduce the introduction of variables into your lab. Some final steps before actually getting your lab ready to open is make sure you've off-gassed enough, test your air if you can, make sure your incubators are up to date, and then go ahead and do an MEA if you can. In our lab, we would use a less complex media like, say, HTF. We use one-cell embryos, and we would turn the oxygen off in our big box incubator to increase stress. We would set up dishes 2 ways, with oil and without oil, and we would open the incubator daily, mimicking procedures. And this, in our opinion, is going to give us the best indicator of a lab air environment. For your benchtop incubators, you can, again, do the same thing without oil, some with oil, open the incubators. And your endpoints should be, though, well-developed, expanded, and hatching blastocyst robust embryos. If you can, try to do some cell count. But I beg you, please don't start up until the lab passes your standards. So how to reopen the lab now? We're going to touch just a couple slides on this. You really need to develop a social distancing program. You want to rearrange your waiting room. And you really want to think about using the parking lot as your initial waiting room. You want to check temperatures on patients before they come in. And our policy is you must wear PPE, no exceptions. And something that's really important is going to dictate based on your individual needs are what's the amount of staff that you can have. And this is going to be impacted by your air exchanges, because we know that the base reproduction rate, in other words, how many people get infected when we don't practice social distancing, with the SARS-CoV-2, it's about 3 to 5 people. Implement cleaning strategies for your exam rooms, common areas, and develop a schedule that's going to reduce or minimize patient-staff contact. Once you kind of got that in place, how do you open up the clinic itself? I would suggest staggering your IVF cases. Start slow, work your way up. Frozen embryo transfers are a really good way to start. All staff have to wear PPEs. Make sure that you use the waiting room and even the parking lot to stagger areas of congestion like the ultrasound rooms and your blood draws, and clean between cases. And ideally, your shift change should be clinic-wide, although that can be really hard to do. Once the staff is, everybody's ready to go, all staff should really wear clean scrubs daily, incorporate sticky mats, shoe covers, masks, open up centrifuge samples under the hood just to deal with the aerosol. And really, I want to implore to you that you can continue to do your embryo procedures as much as you want, because if you think about it, the way we wash gametes, the transfer rate is essentially nothing. And then establish your guidelines for transfer, whether you freeze all or you're going to do transfers. So in conclusion, we really live in an unprecedented time. We don't have all the answers for this. In fact, a lot of these processes we're developing as we go. Again, I want to get back to you though. Cleaning has long-term consequences. Avoid glutaraldehydes. I don't believe that viruses are a big issue for our gametes, because the way we wash sperm samples today in gametes, the dilution factor I think is such that it's not going to have an impact. I do think we should be cautious with sperm samples. Should we quarantine sperm oocyte embryos? I don't know, maybe so. But I do want to tell you this, as an embryologist, don't open your lab until it passes at least one bioassay, because remember, the buck stops with you as an embryologist. So, because when the dust settles, you're going to be the person who's going to have to answer to the medical director and all the patients that your lab is ready to go. So I've got some suggested readings here. The AAB had some really great guidelines, and then all these other guidelines about viral hepatitis, assisted reproduction. And then I've included quite a few guidelines that are used commonly in the tissue culture world. So you can find those here. And then last but not least, I want to thank Tim Schimmel, Antonia Gilligan, and Corey Kelly for all the help that they did in helping me get this presentation together. I have to acknowledge the wonderful laboratory, medical, and most amazing nursing staff that we have here at Palma Fertility. And thank you so much for all your time and effort. Thanks for being here, everybody.
Thank you so much, Klaus, for the wonderful presentation. Very useful. Even someone who is not in embryology like me understands this very well. So I'm sure it has been very clear for other embryologists to understand. We're gonna get the panel joined. So we have with us Dmitri, and we have Levent, both of them. Are you there?
Yep.
Perfect. If you can turn on your camera, that would be good. Perfect. Excellent. Levent?
Yeah, I'm trying. Here you go.
Oh, fantastic. Very good. So I will start with a very simple question, and then we can kind of touch base on the presentation and beyond. The question that everybody trying to figure out is how other labs are doing. Are they operating? Are they holding? Can you tell us a little bit about your experience, your laboratory?
Well, we actually live in the epicenter of where all this went down. So what we had done when ASRM had sent out their notice, we were actually in the middle of treating patients. So we stopped, we did not actively start any more patients in STEM. What we did is we finished the cases that we did, and then we shut down. And we did that mostly to see what the backdrop was going to do. In other words, what was ASRM going to come forward with? But we also wanted to see what was going to happen in Seattle before we opened back up.
Very good. Dmitri?
What is the question?
Is your laboratory operating right now?
I see. No, we're shut down for the last 6 weeks, but we're open and we'll start with the frozen transfers, as Klaus suggested. and our first patients will be here for transfer in about 2 weeks.
Very good. And Levent, you are managing multiple laboratories. Can you tell us a little bit about your operation?
Yeah, after the ASRM guidelines released and we finished all the patients in cycle and we closed down, so we are closed down.
And are you guys planning to open anytime soon? What's your...
Well, we are planning to open probably third week of May.
Yeah. How about you, Klaus?
We are starting stimulation so that we will start retrievals the week of May 17th.
Dmitri?
Yeah. Wait a minute. Well, I did say that we start patients in about 2 weeks. Yeah. So, can I have ask a question for Klaus?
Sure.
Just to follow up. Klaus, I wonder, how do you suggest we treat these patients when they come in if they have antibodies for COVID-19? Do we look at them as patients who would have HIV or hepatitis C, or we just pretty much ignore the status of with antibodies?
That's an awesome question. I think at this point in time, we're probably going to ignore that, that the presence of antibodies or not. Although, because right now at this point in time, we haven't found any evidence of virus anywhere else, in the reproductive tract or in seminal fluids and all that. So a patient who tests positive, we're going to assume is less likely to become infected again, although that possibility is there. But I don't think we're going to treat their gametes any different.
Okay, thank you.
What about you guys?
Well, we'll do whatever the recommendation is, but the concern is what if patients begin to come before there is a recommendation and advise about it, and that's why I was wondering what is your plan. So you will disregard the antibodies?
Yeah.
Okay.
Switching the topic, we'll talk about the ACE2 receptor that was published. I know that Dmitri has a very personal opinion about that. The publication that mentioned the expression of coronavirus COVID-19 receptor in embryos and in sperm samples. What do you guys make of that?
I don't know if these are, and again, I'm just going way out on a limb when I say this. If it's found in developing tissue, you just wonder, is this a receptor site that's universal in developing tissue? And then as the tissue develops, it goes to its end result, like, for example, a spermatocyte, that it would lose this receptor site? That's I'm just throwing that out there.
Sure. Levent?
I don't have any idea. I'm sorry. I don't know. There is not enough research that we can make any sense of out of it, right?
Sure. How about you, Levent, Dmitri?
Well, I have no opinion. I don't think we have enough scientific data to make any qualified statement about this. And I would be very careful about people who jump into the fray to make some kind of sensational claim about anything related to reproduction and COVID virus. It's just not enough time, not enough patience, not enough data. It may take years before we know.
Yeah.
I have an opinion.
The only thing I would add is on the paper published by Klaus on embryo receptors, it wasn't actually detecting the receptor. It was just looking at gene expression, yes. RNA level. So RNA level does not necessarily translate into receptor. So the statements that they made that the embryo has all the machinery to be a receptor for for COVID-19 is not true because it was just RNA level. And gene can be expressed without having a receptor. I want to touch base on the first one of the recommendation that Klaus made, which is the HVAC closed system. And you said that closed system is much better when it comes to dealing with viruses, and that for some laboratories will involve a substantially cost, a change that will cost them a little bit of budgets. What do you recommend for those laboratories who have an open system?
Well, that's a great question. What we've done in several laboratories is if you looked at an architectural diagram of how most labs that are, say in an office setting, there's one set of ductwork that goes off into the embryology lab. A really great way to fix that is if at that point where that ductwork goes off, you can install an auxiliary fan motor. And by installing an auxiliary fan motor at that point in time, what you can do is you can push a lot more air through that laboratory or the procedure room. Now, there's not if you have a drop down ceiling, you're never really going to have positive pressure. But at least if you're able to increase the positive pressure and the air exchanges, so rebalance it so the laboratory has the highest positive pressure, and then as you leave the lab, the positive pressure drops, but still your procedure room and lab would be positive to the rest of the facility. We've done that, and that's worked really well. You can do that in combination with some of these filter systems that you can put inside the lab, like these Xander air units or Coda towers or various things like that. A combination of those things would work, but you can put in an auxiliary fan motor.
Very good. So as you mentioned in your presentation, you're suggesting that the IVF lab would have to upgrade the PPE, and we're talking about very heavy PPE, masks, and maybe a shell, face shield protective mask, or and a lot of other things. This is not something that IVF lab is used to. Are you suggesting? Do you see this implemented in other laboratories as a standard procedure?
I didn't mean that we were going to have to use the face shields and things like that. What I meant more was that we're going to have to wear hair gowns, hair nets, gloves, scrubs, and masks in all IVF labs. And you'd be surprised how many IVF labs people just wear street clothes in, or just don't wear shoe covers in at all. They just have a pair of tennis shoes. They don't treat it like a biological clean room. And so what I'm suggesting is it's good practice. I've never seen a lab drop its pregnancy rates when you increase your cleanliness, whether that's personal to how you keep your whole lab.
Levent, what's your opinion about that?
I agree with Klaus. I don't think that shields are necessary, but anyway, we have to apply the universal precautions for our laboratories anyway, right? We have to deal with the Hep B, Hep C, and HIV patients all the time. So we are protecting, or we need to protect ourselves for those kind of patients. I don't know, we need to take extra precautions because of COVID-19. So we have to do what we have to do, similar like HIV patients or Hep B patients or Hep C patients. If we are scheduling those patients, last patients, maybe we should schedule those patients, COVID-19 patients, the last patients. Similar scheduling. And I think that we have to apply all the universal precautions as necessary. And, I don't think that I'm going to change anything, or I'm going to add any extra steps that I've been using and I'm doing in the laboratory.
Dmitri?
Yeah, it's, as I said, it's so difficult to say what special measures must be taken with COVID. It seems like we're pretty much doing the same what we always do. We have to always assume that patients are infected. You know, that's how you work with tissues. This doesn't change. Even if somebody is negative for antibodies, you should still treat this individual or in any bloodborne, you know, or any fluids as a potential source of contamination. But it's what I'm trying to say. It's all still very speculative. We don't have good scientific data on what additional steps we need to take for coronavirus.
Okay.
If I could add, I think inside the laboratory, we're not going to see a lot of changes because we're so good at rinsing and washing and doing stuff. That by itself will dilute out any potential contaminants. Because we know in sperm samples and when we do an egg retrieval and various things like that, we know that those are not sterile techniques. We know there's a certain amount of carryover when we do that. But the very processes that we do, you know, by washing and diluting and whatnot, really, you know, reduces contamination to virtually nothing right now in the labs. And I think as long as we do that, I think the big thing here is, can we clean our labs better than we were in the past? Do we take this opportunity to be more cognizant of how well we should clean outside? What this is going to really impact this virus now, though, is outside the laboratory, the waiting rooms, the ultrasound rooms, all of those things are going to have, I think, a much bigger impact on, say, nurses and physicians than it will on us, for example.
Yeah. In a previous presentation that was given by Kim Pomeroy, he said that we have dealt with viruses many, many years, and this is nothing different from other viruses we have handled in the lab. And he suggested serial dilutions to minimize contamination. In your presentation, you mentioned the pores of the Zona being approximately 200 nanometer, and the shape of the coronavirus, or the size being approximately 120, making it difficult to enter. Do you, like in a laboratory procedure, would you handle the COVID potential sample that is, you know, when you have a sample, a semen sample, you don't know if it's a non-positive sample or is a positive sample. You're saying that basically, beside the cleaning and all the other precautions, you're going to handle it just like any other sample, right?
Exactly. What we will do now, though, is because we've been reading a little bit more about how to clean, we're going to be much more cognizant to open up all the sperm samples, the vials, you know, the centrifuge tubes under the hood, you know, where you've got the protective plate in front of you, because you have that theoretical aerosol. But other than that, I don't see anything changing.
Very good. Riley, are you there? Riley? Very good. Let's take the poll. If you can release the poll and the questions, let's get questions from the audience.
Of course.
And then maybe we can ask our panel.
Of course. Okay. So we're going to launch the poll questions right now. And while the audience is taking a minute to answer that, I'll be asking some questions from the audience. So the first question is, can the virus be found in follicular fluid and semen samples?
To my knowledge, I've never heard of the virus being found in either follicular fluid or semen samples. And I mostly think that that's because there are no receptor sites in either of those substrates.
What's the other members of the panel think about this?
I have no opinion. I have no opinion. You know, I'm still of an opinion that we need more time and more data. There was no focused or randomized or anything which would be considered evidence-based medicine quality data. And until we have those, I would have no opinion.
Yeah, I agree with Dmitri and Klaus. I mean, there is no indication that I'm not aware of, at least, coronavirus is in the follicular fluid or sperm.
Very good.
Okay, so next question is from Angela, and it's, should we be washing all embryos as a precaution?
Well, embryos let me try to answer this like this. So I'm assuming that the person's asking, should we be doing extra rinsing of the embryos before we transfer them? I don't really think we should, because when the fertilized oocyte, the zygote, was moved to culture, that embryo, or that zygote, I should say, at the time was rinsed several times before you moved it into culture. And then now you're moving it into your transfer media. So I think that you would probably be okay from that perspective. Where I think maybe later on we might become important is if we actually find that this virus is present, then we're probably going to we may potentially have to use some enzymatic solutions, like a serial rinsing through, say, trypsin, for example, before we do a transfer or before we freezing. But I think with fresh transfers, with the data that we have, I think rinsing the eggs after they're fertilized is plenty good.
All right, next question is from Mareed. It says H2O2 is considered embryo toxic. Do you recommend its routine use, and how about OO-safe?
Yeah, actually, hydrogen peroxide is not. I beg to differ with you. It is not embryo toxic. Actually, if you leave hydrogen peroxide just sitting in your lab or out, what it does is when it breaks down, it turns into water and oxygen. So those are two things that are okay to have in your lab. I think what you're referring to is you would not want hydrogen peroxide to come directly in contact with any gametes. Absolutely, they would be terrible. Stuff like O-Safe is a wonderful product, but O-Safe, I believe its main active ingredient is hydrogen peroxide. It just happens to have a stabilizer in it.
Levent, I feel like you want to add something to it.
Yeah, I mean, I don't think that hydrogen peroxide is embryo toxic. We've been using for some time. I don't know, maybe a couple of years, and we did not see any harmful effect on the embryos or sperm.
Very good. There is a question that was directed to Dmitri. Mara, would you mind reading that question?
Yes, it's from Joshua. It says, Dmitri says that his facility is starting with FET. Are they using special consents for the patients? And also, do they feel more comfortable with FET than egg or embryo freezing and placing patients in a holding pattern until we learn more about the effects of the virus on growing fetuses?
Can you please read it again, the question?
Let's break it down into sub-questions so that he can handle one by one.
Sure. So you mentioned that your facility is starting to use, starting with FET. Are you using any special consents for your patients?
We don't use any special consents for patients for FET. We do not.
How about the rest of the panel? Do you guys use any consents different from the current consents?
No.
I heard there was some recommendation. I don't know if it's ASRM or another organization have brought a new proposal for new consents. Klaus, do you know anything about that?
We actually have a consent that was recommended. I'm not sure how our medical director got it, but it's a consent that came that basically states that, in this time, that if you were to develop symptoms while you're in treatment, we would have to cancel your cycle. So basically,
yes.
Basically, the consent is stating that you're about to initiate IVF treatment. You acknowledge that this is all going around us in the background. If you get sick before your egg retrieval, we may not be able to do your egg retrieval. If you get sick after your egg retrieval, we may not be able to transfer embryos. So it's what it's doing is it's forcing the patient to acknowledge some of the risks that are involved in doing an egg retrieval right now. But we're not signing anything for FETs that I know of.
Yes, the question was about FETs, and so it seems like we're on the same page.
Yeah.
There is nothing special. So what was the second part of the question?
Do you feel more comfortable with FET than egg or embryo freezing and placing patients in a holding pattern until we learn more about the effects of the virus on growing fetuses?
I see. So the question is whether we should completely stop transfers and just do retrievals and not transfer anything until we know that the virus doesn't have any teratogenic properties. I guess that's my understanding of the question. And I don't believe so, just because as far as I know, and maybe other panelists will correct me, but I'm not aware of any known teratogenic properties of coronavirus. You know, just as Klaus said, we have to always remember that we do not work in a sterile environment. We work clean, but not sterile. So we have ton of all kind of viruses and bacteria in our environment, and we just take reasonable precautions to prevent contamination, which mostly comes realistically from toxicity comes from oil, and contamination comes from sperm. And so we don't work sterile, and I don't see it unless we find out that this particular virus has teratogenic properties, we should change our practice.
There is a question from Francisco Pereira. He was asking Klaus, because they are freezing a lot of gametes and embryos, if there is any way to use control, some sort of screening of, I don't know what he is referring by screening. Is that a COVID-19 screening or some other screenings?
Well, I mean, as far as the embryos go, what we're going to do, and again, this is probably hypervigilant, too much. We don't need to do it. It's creating unnecessary work. We're just keeping all the embryos that have been created during this time separately until we get some kind of statement from ASRM, or if we don't get a statement from ASRM, then everything will move back into the general in with the general population. I think though with Francisco might be asking also though is, if a patient, what kind of control? So obviously, if you could screen the patients for antibodies or the presence of disease before you initiated treatment after you froze the embryos, that would be amazing if we could do that, which you guys obviously can do for all of us. You could screen our patients and make sure they're okay.
Yeah, no, absolutely.
And I think, screening is going to be both two ends. Do we screen patients up front to confirm we don't have disease or antibodies? Or is there going to be something to help us screen embryos? And I don't see us having to screen embryos. I really don't.
Yeah, exactly. It would be too much.
Yeah.
Then we have to screen them for more than coronavirus.
Oh, yeah. Yeah. Influenza, all kinds of stuff.
Yes, there is no end.
Yeah.
So what do you guys see how do you think this is going to last? Now, looking at the experience of China, Wuhan, and what's going on now with the, the curve in the US, what is your projection? What's your, what do you think is going to happen? Is this going to take a year or months, or are we going to be for this in a new lifestyle? What do you guys think?
I personally think we're in kind of a new lifestyle for a little bit, but it's kind of like what we were talking about earlier. Now, when we first were on this before the call, we've all decided that driving 60 miles an hour is safe, but we're willing to accept the fatalities that go with that. And so we're willing to accept those risks as a society. Now that we have this virus that's going to be living with us probably forever, we're going to have to start thinking about like, what kind of risks as a society do we want to accept? You know, how do we start opening things back up? In order to go back to quote, whatever normal is, there's risks that we're going to have to accept. And I don't really know what the answer is, because I think it's going to vary so much. I think it's going to vary by region of the country. It's going to even vary within the states. Like, for example, Eastern Washington is much more conservative than Western Washington. So I know that the views on coronavirus and in shelter are completely opposite depending on what side of the state you're on. So I can't even imagine how this is going to flow into how we act as a society.
Yeah, and the country is split. Like I look in the neighborhood app and you have 50-50, you know, so half of the people say, you know, I'll stay forever, you know, just to be safe. Half want to go back to life.
Yeah.
So it's, I can tell you one thing, I'm not going to be asked when to open. So, and I have no opinion.
Yeah, how about you, Levent? What do you think?
I think that as I mentioned earlier, I think that we need to learn how to live with it, right? I mean, if you look at Wuhan right now, people are continuing their normal life, and they developed a system to track this virus, or where people go, and then how do they interact each other and everything. So probably we are going to develop similar system all over the world, and everybody is going to track each other. So we need to learn how to deal with this virus, and we need to learn how to live with it.
Yeah, yeah, absolutely, absolutely. So Mara, do we have any other questions, or do you want, or maybe I can start one more question?
We could ask one more question.
Okay.
Do you want to choose it?
No, you can take one of the questions that were asked at the very beginning. I think you missed one or two there.
Okay, this question is from David. It's, what is your plan for when a patient develops symptoms during stimulation up to the day of retrieval?
That's a good question.
I don't know. No, that's a great question. In other words, just to make sure I understand the question, a patient starts follicular stimulation, she's starting to go through stimulation, and now she shows symptoms. Is that? And what are we going to do with patients like that? You know, luckily, if the patient is early enough in the follicular stimulation, our hopes are that we can just down-regulate the patient and, you know, just stop the egg retrieval, not even do the egg retrieval, cancel. Something that I'm more concerned about is, what if you have a patient who's got, you know, 10 follicles on each ovary, they're all, you know, 14, 18 millimeter follicle, she's ready to trigger, all of a sudden she's sick. To not do an egg retrieval on that patient could be a real issue. You could create, you know, a cystic ovarian situation that could be terrible. So that's where I really want the physician input on what should we do with those kind of patients. Do we do the egg retrieval in a kind of a quarantine kind of mode, freeze the eggs and then clean the lab? What should we do there? I could use some guidance on that aspect.
Sure. Dmitri?
I mean, I guess we have a framework for embryology, right? So we're not practicing medicine, and as far as embryology, I think that if a physician decided it's safe to retrieve the eggs, we just treat them as in egg retrieval. There will be no, I can't imagine any impact of patient general, like signs of infection, such as, you know, fever or something, and quality of the eggs. You know, we just did not see anything like that. So once decision is made to go ahead with egg retrieval, I don't see any issues with embryologists just, you know, doing it as they always do,
okay,
without any changes.
Yeah, Levent?
I agree with Dmitri. I mean, if this is a medical decision, a physician, treating physician make the final decision. If they go ahead and do the egg retrieval, we are gonna do the things in the laboratory and either egg freezing or continue the embryo. And I think that it's gonna be a medical decision. It's not, I don't think that we are gonna be having any kind of influence on the medical decision.
Sure. Let's go to the poll. Mara, if you can give us an idea what the poll looks like.
Yeah, so we asked the audience, are you currently operating your IVF laboratory? And 53% said that they are not operating. 41% said they're minimally operating, and 5% that said they're fully operating. We also asked, did this webinar help you prepare for the opening of your IVF laboratory? 92% said yes, 1% said no, and 8% said not sure.
That's pretty good.
That's us, right? You did a pretty good job there.
Thanks, brother.
Next question is, what actions would you consider implementing during your lab opening? 92% said cleaning techniques, 31% said HVAC systems, 20% said upgrading their laboratory. And the last question we asked, would you be using any screening method to reduce the risk of COVID-19 exposure and lab contamination? 78% said screening patient and staff for temperature, 23% said screening using real-time PCR, 15% said screening using antibodies, and 19% said all of the above.
Very good. So what do you guys think about the poll?
Klaus did a good job. Great job.
Happy.
I'm impressed.
I've been happy. Thank you, guys. Y'all are too kind. It's only because it's my birthday that you cut me some slack, right?
So how about the question about how do you screen your patients? We still see the majority wanted to screen using, temperature and monitoring system for the patients. I've looked at one study from China yesterday. They had multiple papers looking at symptoms of patients. I think 86 or 85% of the patient did have fever. So it's a pretty good starting triage, right? That you could do for your patients before they get enrolled.
Let me ask you this, Nabil. How accurate this test?
The test. So it was a whole presentation.
I mean, honest opinion.
Yeah,
how accurate this test are?
Yeah. So if you look at the rapid tests, so the rapid tests are those little cassettes. Yes. Those are according to a publication that we have reviewed and according to experience that Europe has seen, Spain in particular, they are approximately 30% sensitivity. So most of the tests that you see in the market, in terms of false positives, they are not bad. They're pretty good in determining when someone is positive. The problem is when someone, the false negative rate is pretty high. That means that, for example, these rapid tests, they have 30% sensitivity, so they miss a lot of positives there. So when
it means that if it's negative, doesn't mean that negative.
That if you use the rapid test.
Yes.
But there are other tests, the ELISA tests, that are medical grade. It involves a lot of, equipment and just a better test, right? Those are in the 90s, 90, 91 or above %. That's on the antibody side. On the real-time PCR side, when you look at the literature, you get a mix bag. You get those ones that are in the 50s, 60s, and you get those ones that are in the 90s, 80s, 90s. And each study is different and each design is different. Now, what we have learned so far is also the collection, specimen collection is important. So when you are collecting, when you do nasopharyngeal, not everybody knows how to do it. If you let the patient try to do collection themselves, most likely they're not going to be going deep in their path, and that can cause some of the negative results, false negative that we see. Also, the timing of specimen collection. If you take someone early on, very early in the stage of infection, you may miss it. If you take them on a week, at least a week, for or 4 or 5 days for viral infection, 2 weeks for IgM, and 4 weeks for IgG. If you don't time it, you may get a false negative as well. But you know, there is no perfect test. That you have to look at the context. Yeah. Very good. So what I would say.
Can I ask you a question?
Yeah, sure.
You know, one of the things that a lot of the, you know, small standalone clinics that, you know, I talk to about just trying to develop some ideas about testing and whatnot like that is the, you know, is the having to send out the test results. You know, I think that's where a lot of bottleneck is occurring right now, and why people are willing to go towards more non-FDA approved type testing.
Yeah. The Abbott test, I don't know the data from that rapid test. I know that there is a shortage, and that the government has taken most of those equipment, and then you have certain hospitals. I don't have data on the Abbotts. I heard, but I don't have anything like, you know, tangible I could discuss. But right. I know that those cassettes are definitely. You know, they're out of control in terms of manufacturing and all this. Like, you find them everywhere. It's more of a consumer product. I see it's more than a laboratory product. Yeah. So that's really good. So I'm gonna ask you a question, Klaus. That I know that you have been involved in helping lots of laboratories, you know, start a new lab. And this, for me, looks like more starting a new lab, right? Because most labs are closed and they have to start again. And are you receiving requests from people that wanted you to help? Are you planning to help some labs?
I've received a few requests, but not very many. I mean, I'd be glad to help labs that need some help, you know. I mean, the way I would look at this is we would just go in and set up a very stringent, you know, protocol based on, you know, your particular situation. But yeah, I mean, these are, the way I would look at this is as a consultant or as a third party looking in is just, what are the ways that you can come in and really tighten up your processes? And this is one of them, because when you go to a lot of laboratories, and I'm not going to mention any names, we've heard the term dust bunnies. I've seen dust jackrabbits in labs. These things are monsters. These things are giant balls of lint that have collected under the hoods because nobody's cleaned their lab in God knows how long. And these are really good opportunities to kind of go in there and re-clean. So if somebody was to email me or call me, I would say, follow the protocols that I've given you. You know, I'd be glad to talk to you any way I can through it. But really, if you just take the approach as I'm just going to really clean my lab very well, and then while I'm down, I'm going to look at all my protocols to see how I can improve them. I think that's where you're really going to see the best use of your time.
Just to add to this, if you guys want access to this presentation, so it will be posted on Progenesis Academy page. All the webinars are there. Feel free to go next day, and then you can look at the video, and then you can take notes of all the slides that Klaus has shared with us. Dmitri, you are currently the president for the American College of Embryology. Tell us a little bit. I don't know if everybody on this audience know about the American College. Can you tell us a little bit what you guys do?
Yeah, I'll be very short. I will just take this opportunity to invite embryologists to join the college. You can go to the college pages embcol.org, or you can search on Google for American College of Embryology. Our goal is to promote embryology to the public, elevate its status, and we're doing this by promoting embryologists. So please go to the website and look up at our site.
Very good. Is there anything you guys wanted to add before we wrap up the webinar?
Thank you guys for taking time. I know so many people are webinared out, you know.
Yeah.
But thanks for doing this. Thank you for the invitation.
Thank you. Thank you. Well, thank you so much for coming and presenting at Progenesis Academy, and for the panel, Dmitri and Levent. Thank you so much for your participation and advice. And you, thanks to Progenesis Academy for hosting this seminar. Next week, we will have another webinar. Stay tuned, check your email. Thank you so much, guys. I hope you have a great day. Thank you so much for your participation.
Thank you.
Thank you, Nabil. Bye-bye. Thank you, Klaus.
Thank you,
Thank you,
guys.
Levent.
All the
Bye-bye.
best, everybody.
Bye-bye.
Be safe out there.
Bye-bye.
Bye.